Hi all. In this video, I am going to discuss about structure and reproduction of Aspergillus species. In this, we are going to see some species of Aspergillus. They are Aspergillus niger, Aspergillus flavus, Aspergillus fumigatus and Aspergillus tertius. Next, we are going to see about taxonomic position. Kingdom fungi, division Ascomycota, class Uroshiomycetes, order Uroshiales, genus Aspergillus. Aspergillus, commonly known as black mold, is represented by about 100 species which are widely distributed from Arctic to tropical regions. Species of Aspergillus grow as contaminant in the laboratory cultures as their spores are present in the air. A large number of species have been found in the soil. Majority of the species are saprophytic and grow on decomposing organic substances such as fruits, vegetables, jams, cheese, wood, leather, etc. A few species are parasitic and cause disease of animals and human beings. Aspergillus can be grown easily by keeping a piece of cheese or bread in a warm, moist chamber. Vegetative structure of Aspergillus. The body is mycelial. The mycelium consists of slender, tubular, pale-colored, extensively branched, thin-walled hyphae. Some hyphae ramify superficially upon the substratum while some penetrate into the substratum to absorb the food material. Each cell is multinucleate and is filled with granular cytoplasm, mitochondria, endoplasmic reticulum, ribosomes and vacuoles. The crosswalls between the cells have a simple pore through which the cytoplasm of the adjacent cells remain continuous. Reserve food material is in the form of oil globules. In this slide, you can observe the structure of Aspergillus species. Reproduction in Aspergillus is of two types, asexual and sexual. Asexual reproduction by the production of conidia. That is, chain of conidia we can observe in Aspergillus species. The conidiophore is arising from a food cell. Each food cell produces a special erect branch as an outgrowth. It is a young conidiophore. The tip of the conidiophore swells up into an elliptical or globular multinucleate head called vesicle. It forms many radially arranged tubular outgrowths called sterigmata or phyllets. In some species of the Aspergillus, we can see uniseriate sterigmata, and in some species, secondary sterigmata also will be present. If it is like that, we can tell them as biseriate. Conidia arise exogenously from the sterigmata or phyllets. Therefore, conidia are also called phyllospores or phyloconidia by abstraction method. They are arranged in basipetal succession, that is, the youngest conidium is at its base and the oldest at the tip. The sterigmata elongate at the tip to form a tube. The conidia are formed inside this. The sterigmata are uninucleate. At the time of formation of conidia, the single nucleus of the phyllid divide mitotically into two daughter nuclei. One of the daughter nuclei passes into the tube. It is the first conidium. As the first conidium is formed, the upper broken wall of the phyllid serves as a cap around it. The second conidium is formed by phyllid just below the first. The cytoplasm of both the conidia is confluent through a narrow cellular link called isthmus. The continuity of the cytoplasm is stopped by the formation of the inner conidial wall. The isthmus becomes empty and now it is called connective. Conidia are small, globose, unicellular, uninucleate or multinucleate, black, brown or yellow-green in color. They have two-layered wall. Outer wall layer is thick, spiny, pigmented and known as epispore, whereas the inner one is thin, delicate and is called endospore. Conidia are dispersed by wind. They germinate on suitable substratum by giving out a germ tube. The germ tube becomes septate, branched, and forms a mycelium. Here you can observe the conidia formation. Next one is sexual reproduction. Majority of the species of Aspergillus are homothallic. However, a few species are heterothallic. It takes place by the formation of male and female sex organs. Male sex organ is known as antheridium. Female sex organ is called aspergonium. The tip of the archegonium arches over aspergonium and fuses with it. The wall at the point of contact dissolves, thus making a continuous passage. It is plasmogamy. The contents of the antheridium pass into the aspergonium. The pairing of male and female nuclei takes place in the entire structure is known as develops into a fruiting body called mini-asci. After the pairing of the nuclei, the aspergonium becomes septate. Is known as segment consists of one male and one female nucleus. Germ tubes develop from each ascospore. From the ascospores, mycelium develops. The nuclei in these cells fuse to form diploid nucleus, that is karyogamy. The diploid nucleus first divides meiotically and forms 4 haploid nuclei. Each haploid nucleus divides mitotically and thus 8 haploid nuclei are formed. Each nucleus later on gets surrounded by cytoplasm and develops a wall. Thus, 8 haploid ascospores are formed in each ascus. Some species of Aspergillus produce antifungal and antibacterial compounds. Aspergillus niger is used to treat the soil for the tracing out of elements like copper. It can detect copper even in traces. Aspergillus niger can use for the production of organic acids such as citric acid, gluconic acid, and gallic acid. Aspergillus terreus is used to produce itaconic acid. Aspergillus flavus and Aspergillus oryzae is used to produce kojic acid. Many species of Aspergillus are good sources of vitamins such as riboflavin. Aspergillus can produce enzymes such as amylase. Aspergillus oryzae is used for fermenting rice to produce wine and in making miso and soja sauces. Aspergillus is saprophytic in nature, so it can decompose organic matter. Harmful activities of Aspergillus: spoilage of food and other articles. Aspergillosis disease to humans. Aspergillus niger causes fruit rot of pomegranate, mango, etc. Aspergillus flavus produces aflatoxin. It's a cancerous compound.